Well, it's time to begin our discussion on Chapter 8, Neoplasia. This 30-page plus chapter provides us with a lot of information, but of course there will likely be other supplemental web sources I will post for you on Blackboard. We'll start out with concepts of cell differentiation and growth. I'll discuss the cell cycle, and I will introduce to you cyclins and cyclin-dependent kinases, or CDKs, and cyclin-dependent kinase inhibitors, CDKIs. I'll discuss the difference between cell differentiation and cell proliferation. Then I will transition into characteristics of benign and malignant neoplasms. I'll talk about how we name them and the difference between benign and malignant neoplasms. Next, I'll discuss the etiology of cancer, and I will focus on cancer-associated genes, but I will briefly discuss epigenetic mechanisms and the immune response that is involved in cancer. The section on the etiology of cancer is highly redundant, and you should keep in mind that rarely is there just one single cause of a particular type of cancer. For example, roughly 10% of breast cancers have a genetic cause, the rest are sporadic, and finding the etiology is more difficult. Development of cancer is generally thought of as a multiple hit process where a person initially has a pre-existing genetic component that makes them more susceptible to cancer, and then you add in other risk factors like exposures to different chemicals, carcinogens, exposure to viruses, radiation, etc. Next, I'll discuss clinical manifestations we would expect to see in a patient with cancer. Topics like anorexia and cachexia and manifestation of sleep disorders will be discussed. Moving through the chapter, next I'll discuss our diagnostic methods to grade and stage tumors. I'll also talk about tumor markers and how we use them. This is a topic I love as it reminds me so much of my fellow graduate students' research topics in cancer. We use these tumor markers, which are really antigens from the cancer cells, for monitoring their decrease in levels as we treat the cancer. I will follow up this topic with treatment of cancer, of course. The go-to standard is surgery, but there are other interesting treatments to discuss as well. If I can find something on the web as a resource on childhood cancers, I will post it. Most of my dissertation used a cancer cell line from 2 children that died from a pheochromocytoma. Your book isn't too interesting with respect to this topic, in my humble opinion, so I'll try and find something comparable on the web. At least, I hope to do so. The question is, are we winning the war against cancer? I always joke with my traditional face-to-face students that if we were mice, we would have the cure to most cancers already. Scientists have discovered drugs and treatments that cure different cancers in mice, but for whatever reason, their effectiveness does not transcend into humans. But we are still winning that war against cancer. Every day we are developing new forms of chemotherapy or biotherapy. Cancer is not the death sentence it once was. The 5-year survival rate has improved drastically since the 1960s. Cancer is not a single disease. Cancer in the prostate behaves differently than pancreatic cancer. The most common form of cancer for men is prostate cancer, and the most common for women is breast cancer. Let's start off simple. What is the difference between dysplasia versus neoplasia? Well, I'll remind you from our earlier lectures that dysplasia involves cells that have changed in size, shape, and organization. Hyperplasia involves abnormal proliferation of cells, but that should stop once the stressor or causative agent is removed. Neoplasia differs from the other two because this now involves disorder of altered cell differentiation and growth. The new growth tends to be crab-like because cancers grow and spread by sending crab-like projections into the surrounding tissues. So, neoplasia is an abnormality of cell growth and multiplication and it is characterized by excessive cellular proliferation. Even after the stimulus is gone, there is uncoordinated growth and there is tissue infiltration. At the molecular level, there are disruptions of the growth regulatory genes, and all of this develops in a multi-step fashion. As I continue to lecture, pay attention for these regulatory genes when we address the cell cycle. Be ready. We are going into more detail for the cell cycle than probably what you learned in an introductory biology or anatomy or even microbiology class. I would have to say your textbook does a good job talking about proliferation versus differentiation, and it does a good job of reminding us about programmed cell death, or apoptosis. With cell proliferation, we are talking about the replacement of cells or the generation of new cells. It speaks to mitosis. Cell differentiation is the selection of genes to be transcribed. That means forming a working copy or transcript or messenger RNA. And then the mRNA is translated or decoded into a protein. In every nucleated cell of your body, you have the same genome, but not every cell looks the same, nor has the same physiology. A cardiocyte looks and behaves as a cardiocyte, but an osteocyte looks and behaves as an osteocyte. Why? Each cell, as they differentiated, preferentially selected some genes to be expressed and others to be silenced. A cardiocyte would silence bone genes, for example. Cells that are fully differentiated typically have reduced rates of cell proliferation. It's a trade-off. Apoptosis is, of course, a civilized form of cell death. It is clean and orderly. If a cell sustains enough damage to its macromolecules or enough free radical oxidative damage occurs, then a series of events leads to the hydrolysis of DNA, packaging of cellular components, and then a macrophage phagocytizes the cellular debris. It is time to address the cell cycle, or the interval between each cell division. The cell exists in a phase known as interphase, and that's the longest phase. Interphase has three subcategories, G1, S, and G2. The cell cycle begins with G1, which stands for gap 1. Cellular activities during G1 involve preparations for DNA replication, which will occur during S phase. There is increased protein synthesis and synthesis of organelles and cytoskeletal elements. The S phase stands for synthesis phase. This can last 10 to 12 hours, and during this precarious time, the chromosomes are being replicated. Each strand is unwound by a helicase, and each strand serves as a template for a new daughter strand. This process is referred to as semi-conservative replication. After S phase, there is a second gap phase, or G2. This is called the pre-mitotic phase. Enzymes that are needed for cell division are synthesized, and this involves gene expression. The DNA is checked for errors and damage that might have occurred during replication. M phase is the time when the nuclear DNA is divided between two daughter cells, and also it has a component known as cytokinesis when the cytoplasmic contents are split between the two daughter cells. Perhaps you are recalling terms involved in mitosis like prophase, metaphase, anaphase, telophase, and cytokinesis. In some cells, they may enter a special phase known as G0 or gap 0. I always learned it in grad school as G0, but I will use the term gap 0. In this case, a cell leaves the cell cycle temporarily or permanently. It exits the cell at G1 and is often called quiescent, but that implies that the cell is inactive, and that is most certainly not the case. The cells are busy carrying out their functions in the organism. Often G0 cells are terminally differentiated. They will never re-enter the cell cycle, but instead They will carry out their function in the organism until they die. For an example, neurons. For other cells, G0 can be followed by re-entry into the cell cycle. Most of the lymphocytes in human blood are in G0. However, with proper stimulation, such as encountering the appropriate antigen, they can be stimulated to re-enter the cell cycle at G1 and proceed on to new rounds of alternating S phases and mitosis. G0 represents not simply the absence of signals for mitosis, but an active repression of the genes needed for mitosis. Cancer cells cannot enter G0 and are destined to repeat the cell cycle indefinitely. Before the cell moves from one phase to the next, there are checkpoints. For example, there is a checkpoint between G1 and S phase known as the G1S checkpoint, sometimes referred to as restriction point 1. And there is a checkpoint between G2 and mitosis known as the G2M checkpoint or restriction point 2. At G1S, the checkpoint involves proteins that check the DNA for any damage that should be repaired prior to replication. For example, uracil is found in DNA. It can result from spontaneous deamination of cytosine. This uracil, if not repaired, can cause a point mutation when the DNA is replicated. The second checkpoint will check for any DNA that wasn't replicated, and it will also be checked for any replication errors. In the next few slides, I will identify proteins called cyclins. There are many types, and these are expressed at specific times of the cell cycle. The cyclins control the entry and progression of the cell through the various phases. Cyclins interact with CDKs to activate them. A kinase is an enzyme that attaches a phosphate group to other proteins. Proteins that are phosphorylated have a change in their activity. CDKs, or cyclin-dependent kinases, unlike cyclins, are constitutively expressed. They are all in the cell all the time. The idea is they are not activated until their cyclin partner is expressed. We will also see antagonistic proteins called CDK inhibitors. Really, it all comes down to maintaining a series of checks and balances to ensure proper cell growth and division. Let's go through the cell cycle again, but this time let's focus on the cyclins and cyclin-dependent kinases and cyclin-dependent kinase inhibitors I introduced on the previous slide. G1 cyclins involve D cyclins. These are not shown in your book. The S-phase cyclins are E and A, and mitotic cyclins are B cyclins. Again, their levels rise and fall with the stages of the cell cycle. Cyclin-dependent kinases include a G1 CDK known as CDK4, an S-phase CDK known as CDK2, and an M-phase CDK known as CDK1. Their levels in the cell remain fairly stable, but each must bind the appropriate cyclin, whose levels fluctuate, in order to be activated. The CDKs add phosphate groups to a variety of protein substrates that control processes in the cell cycle. During G1, a rising level of D cyclins bind to CDK4 and signal the cell to prepare the chromosomes for replication. A rising level of S-phase promoting factor, or SPF, which is a complex of A cyclins bound to CDK2, enters the nucleus and prepares the cell to duplicate its DNA. As DNA replication continues, cyclin E is destroyed, and the level of mitotic cyclins begins to rise in G2. The B cyclins combine with CDK1 to form M-phase promoting factor, or MPF, and this initiates the assembly of the mitotic spindle, breakdown of the nuclear envelope, cessation of gene expression, and condensation of the chromosomes. The two pictures on this screen show how these cyclin and CDK complexes form and are subsequently degraded. The cyclin CDK complexes are broken down by the CDK inhibitors. So it really is the formation and accumulation of the cyclin CDK complexes and their subsequent removal by the CDK inhibitors that regulate the cell moving from one phase to the next. And you should keep in mind that new cyclins and CDKs and even CDK inhibitors are being discovered on a daily basis. The cell cycle story is a continuously changing story as we discover more of the chemical signals that regulate it. I like this last picture. It focuses on cyclin B and the synthesis of MPF. And in this graph picture, you see levels of cyclin B and MPF rise in a coordinated manner to promote mitosis, and then their levels drastically decline as the cell enters interphase, only to lead to the accumulation of cyclin and MPF as the cell approaches the next mitotic phase. It is important to note that these cyclin, CDKs, and CDK inhibitors are proteins. Mutations in their genes have been associated with cancer. These genes are considered to be proto-oncogenes, and I'll speak more on this later. This should not be surprising since checkpoint failures would allow the cell to continue to the next phase despite damage or lack of integrity of macromolecules. A perfect example of a failed checkpoint due to a loss of a tumor suppressor gene can be seen in the p53 protein. p53 protein, of course, is the product of the p53 gene. p53 is responsible for checking DNA integrity before DNA replication and p53 can halt progression of the cell cycle in G1 by blocking the activity of CDK2. Both copies of the p53 gene must be mutated in order for this checkpoint to fail. So mutations in p53 are considered to be recessive, that is, both copies must be mutated. And p53 is really seen, like I said, as a tumor suppressor gene. We will have more discussion on tumor suppressor genes later. We have seen p53 in an earlier lecture as it plays a role in the intrinsic pathway of apoptosis. Clearly, if a cell has two mutated p53 genes, the cell will live on and possibly enter a cancer cell cycle because it is never provoked to enter into apoptosis. More than half of all human cancers do in fact harbor p53 mutations and have no functional p53 protein. Your textbook has many pages dedicated to cell proliferation and cell differentiation. Cell differentiation is certainly one of my favorite topics, but I should also discuss cell proliferation. In your body you have about 200 different types of cells. These can be divided into three large groups: well-differentiated cells like neurons and skeletal muscle that rarely divide or reproduce. 2, the progenitor or parent cells that continue to divide and proliferate, like blood cells, skin cells. And 3, the undifferentiated stem cells that can enter the cell cycle and produce the progenitor cells in the body when the need arises. As a stem cell replicates and divides, it will give birth to two daughter cells shown here in the bottom picture. One of the two daughter cells will remain as the stem cell, while the other will become a progenitor cell, which can also replicate and divide to give rise to daughter cells. Each of these daughter cells is becoming increasingly more determined. That means there is selective gene expression, some genes turned on, others kept off. The end result, of course, after a series of maturation steps, is that the cells will become so specialized that they will lose their mitotic abilities. To help you understand this process, I'll give you the same example I give my face-to-face classes. I usually ask the students to raise their hand if they are planning on becoming a nurse, or PA, or OT, or PT, or dental hygienist, etc. After I go through the list and the students take turns raising their hands, I say to them, well, I had to ask because right now you all don't look different to me. yet you are all determined to become something. You won't look different to me until you get into your professional school, complete the courses, take your board examination, and graduate. Then you all will have uniforms and maybe stethoscopes or some other paraphernalia that will let me identify what you have become when I look at you. Not until then will you look different to me. Cells are the same way. They are born one day, make a molecular decision to become a certain type of cell. This means become determined, and they start that process by turning on or off certain genes. But not until they finish this gene selection and express specific proteins that allow them to look a certain way and act a certain way will they become differentiated. There are some words we need to use for stem cells. Not all stem cells are as powerful as others. A totipotent stem cell means that it can develop into a new organism as well as the extra embryonic tissues like amniotic sac, placenta, etc. The best example of this is a zygote. It can generate the 3 embryonic layers as well as generate the trophoblastic cells that will create all the membranes around the new embryo. A pluripotent stem cell can give rise to a new organism but cannot generate the tissues required to attach the embryo to the uterus. For example, if we were to take cells from the inner cell mass of a blastocyst, these could recreate a new organism, but they could not generate the extra embryonic membranes. The best example of a multipotent stem cell is the hemocytoblast. This cell replicates and divides, but only gives off daughter cells that can generate foreign elements in the blood, like platelets, lymphocytes, etc. A multipotent stem cell can replicate and divide to many different daughter cells, but they all have a common function. In this case, they will generate a cell found in the blood. The multipotent stem cell will give rise to 2 types of progenitor cells, either the lymphoid or myeloid progenitor cells. The lymphoid cells will give rise to all T and B cells. The myeloid progenitor cells will give rise to all other types of formed elements, the remaining white blood cells. And that would include platelets, erythrocytes, etc. Now, a unipotent stem cell can replicate and divide, but can only give rise to daughter cells that will differentiate into 1 type of cell. The best example of this are the spermatogonia that replicate and divide but only give rise to sperm. Now it's time to change the tone a little and talk about characteristics of benign and malignant neoplasms. Parenchymal tissue represents the cells in an organ that basically do the function or functions of that organ. In other words, they are the functional components of the organ. For example, in the lung, the parenchymal cells are the simple squamous cells that line the alveoli and allow for gas exchange. Stromal cells are supportive cells. They are responsible for making the extracellular matrix and connective tissue that surround the parenchymal cells. In the stromal tissue, we would find the blood vessels and lymphatic vessels that would provide nourishment and support for the parenchymal cells. The word tumor really means a swelling that could be caused by a number of conditions like inflammation or trauma, but it is also used to define a mass of cells that arise due to overgrowth. Tumor and neoplasm are often used interchangeably, but tumor should be used as a more generic term for swelling, versus neoplasm should be used for new growth due to increased cell number. A benign neoplasm is relatively harmless unless a vital area is involved. Your text addresses benign neoplasms that cause pressure on adjacent structures that lead to ill health, but typically they are relatively harmless. A malignant neoplasm is cancerous. They grow and spread throughout the body. A picture of a benign neoplasm is shown here on the bottom right. You can see they are well defined, they are encapsulated, The cells under histological examination appear similar to the cell of origin. They do not spread to other tissues. They have slow growth and usually not fatal unless they are impinging upon another organ or blood vessel. Malignant neoplasms, as shown here in the top right picture, are very invasive, have vague borders, and the cells are undifferentiated. They appear to be very immature versions of the cell of origin. Metastasis is common where the tumor can spread to other organs through the blood or lymph. They have a high growth rate and mortality. We named the benign neoplasm by adding the suffix -oma to the parenchymal tissue type from which the growth originated. For example, if it's an epithelial tissue of origin, we would call this a papilloma or adenoma. The way we name the malignant neoplasm is a bit more obscure depending on the origin of the neoplasm. Mostly we add the suffix carcinoma, if it is of epithelial origin, that is, or we add sarcoma, if it is derived from connective or muscle tissue. There are specific terms for malignant neoplasms for neural tissue or hematologic neoplasms. As you can see here under the column of benign tumors, the suffix -oma is merely added to the originating tissue type. And again, you can see the use of carcinoma or sarcoma for malignant neoplasms in epithelial or connective tissue / muscle endothelial tissue, respectively. I'd like to talk about malignant neoplasms a bit more. How is there a loss of cell cycle regulation? The cells visually look undifferentiated and they don't resemble the tissue of origin well. They have a high mitotic rate and they break through tissue barriers. They are invasive and can travel through the blood to take up residence in other areas of the body. The access to blood is important so they can keep getting nutrients. There is also genetic instability in the cancerous cells. There can be point mutations in proto-oncogenes that give gain of function, loss of tumor suppressor genes, chromosomal anomalies, and despite the DNA damage, the cells escape crisis and apoptosis. It is also important to note that the surrounding tissue, the stromal cells and extracellular matrix itself, factor into this equation. There are signals that go from stromal cells to the parenchymal cells. The parenchymal cells don't have to be the initial cause of cancer. It can be aberrant signaling from the stromal cells that initiate the cascade we call cancer. The stromal cells give off growth factors and tell parenchymal cells when to grow, how to grow, and when to stop growing. Clearly, aberrant signals from the stroma can lead to cancer of the parenchymal cells. Cancer cells also don't have cell density-dependent inhibition. They can grow on top of each other or even as free-floating cells in cell culture. This is what we mean by cancer cells losing their cell cohesiveness and adhesion. They lose anchorage dependency. Cell-to-cell signaling is aberrant. That's what I was speaking of just now. And you can read the rest. Many of these cancer characteristics are discussed in this week's supplemental videos, and I hope you pay attention to those. The steps required for a primary tumor to metabolize are the following. First, you need the primary neoplasm to replicate and divide. That tumor must be able to break through the extracellular matrix. Cancer cells do this largely by expressing the gene for the type 4 collagenase enzyme, and this allows them to break through the basement membrane of a nearby capillary. This leads to intravasation of cancer cells into the capillary and evasion of immune cells that could destroy them. Many cancer cells will form aggregates with thrombocytes, basically coating themselves with platelets to help with immune system avoidance. Then the tumor cells need to be able to break through a vessel again and take up residence in an area where the microenvironment will support them. This is a process called extravasation. And of course, those cancerous cells need angiogenesis. We all have areas in our body where we have developed tumors. The thing is, they eventually stop growing and are contained because they don't have enough nutrient supply. Read, they don't have a blood supply to deliver nutrients. It's when the cells switch their chemical signaling that triggers angiogenesis that the tumor can now get big enough to be detected. Three primary things will now dictate the rate of tumor growth: number of dividing cells, length of the cell cycle — if shorter, you can double your population size faster — and the number of cells being lost, if any. The growth fraction of tumor cells is greater. Growth fraction refers to the ratio of dividing cells to cells in G0. Cancer cells don't necessarily have a shorter cell cycle. Their rate of growth is usually due to more cells actively dividing rather than going into G0. When we speak of doubling time, this refers to the length of time to double the mass of a neoplasm. In order for a tumor to be detectable, it needs roughly 30 doublings, or needs to be about 1 billion cells big. Let's start to focus now on known causes of cancer. Your textbook separates causes of cancer into two broad categories: genetic and host/environmental factors. We will begin with a focus on the genetic approach to cancer. A proto-oncogene is a normal gene that is found in the nuclear DNA. These genes code for proteins that would promote cell growth and division. These proteins might include growth factors, or receptors for growth factors, or second messenger factors that alter gene transcription and translation of other proteins so that a cell can increase its growth before division. In this picture at the top right, you are seeing a picture of a growth factor binding to a receptor and activating second messenger systems within the cell. Second messengers activate kinases, proteins that phosphorylate other proteins. The example here being mitogen activating protein kinase, or MAPK. And this MAP kinase is shown phosphorylating a protein involved in gene expression, and thus turning on genes that are involved in upregulating mitosis, and that means more cell division. When proto-oncogenes have a mutation that lead to a gain of function, then these genes are overly expressed, and we now call this gene an oncogene, or a gene that promotes cancerous growth. In some cases, the mutation leads to a new protein altogether that leads to aberrant cell growth. In contrast, tumor suppressor genes code for proteins that help downregulate the cell cycle. The two most well-known genes are the retinoblastoma gene, RB, and the p53 gene. These two proteins participate in cell checkpoints, and of course, we remember p53 playing a role in the intrinsic pathway for apoptosis. When tumor suppressor genes are mutated, we are worried about the loss of their function, or more simply, the loss of cell checkpoints to arrest the cell cycle to allow for DNA repair. So I'm hoping you can see the balance between the activity levels of these two types of genes. In a normal cell, they should balance each other. Cancer can occur due to activation of growth-promoting oncogenes, as shown in the picture, or conversely, it can result from inactivation of tumor suppressor genes, or in genes that control apoptosis. All can lead to unregulated cell differentiation and growth, which leads to a malignant neoplasm.